Hi, I'm Tom Clendenin, and this is Carl Winnicke. And we're going to describe experiments that Carl and another graduate student in the lab, Jonathan Leon, did to uncover the computations fruit flies use to process visual motion. Broadly speaking, our paper is about the computations neurons perform to detect specific features of the sensory environment. Because we study vision, we're interested in how neurons detect a particular visual feature, the direction of motion. Neurons that can do this are said to be direction selective, meaning that they respond more strongly to motion in one direction than any other direction. But the visual world enters the brain through neurons that are not direction selective. So that means some neurons must generate their own direction selective signals by receiving and transforming signals from non-direction selective neurons. And it's the nature of this computation that our paper focuses on.
Visual neurons are a powerful model for studying neural computation because we can design visual stimuli, present them to the fly, and measure the neuron's response using genetically encoded voltage or calcium sensors, allowing a direct readout of how visual circuits transform inputs into outputs. And the fruit fly is an excellent organism for studying the visual system because we know so much about it. For example, we have a complete and highly stereotyped wiring diagram of peripheral visual circuits. We know how many visual neurons respond to different inputs, and in some cases, we know what kinds of ion channels and neurotransmitters they use. Since the 1950s, the neural mechanisms underpinning direction selectivity have been a major area of research, inspiring several computational models. These models propose different algorithms for how neurons combine information over space and time to generate direction selective signals. In one model, this information is simply added together during motion in any direction. This is a linear transformation of the input signals, and it generates a direction selective signal that is then nonlinearly transformed to become even more direction selective. Several papers have reported that this is actually what's happening in simple cells in the visual cortex of some vertebrate species such as monkeys, cats, and mice. In flies, however, for decades it's been thought that during motion in the preferred direction, visual signals from different regions of space must be multiplied together, that is, nonlinearly transformed, in order to become direction selective at all. And more recently, the prevailing view has been that a different kind of nonlinear transformation also occurs to suppress responses to motion in the opposite, or null direction. Our paper, however, shows that flies have neurons whose computations strongly resemble those of vertebrate simple cells. That is, direction selectivity in flies can emerge from a simple linear transformation. To show this, we use two-photon imaging to measure changes in membrane potential in one of the two cells where direction selectivity first emerges, a cell called T5. And we show that T5's response to a moving stimulus is direction selective and is equal to the sum of its responses to stationary stimuli that are equivalent to the moving stimulus when added together. And this is a classic way of determining whether a neuron is simply adding inputs across space and time. We believe that the nonlinear signal transformation, previously thought to be necessary for direction selectivity in flies, is actually the transformation within the direction selective neuron from the voltage signal to the intracellular calcium signal, which again resembles vertebrate simple cells, where a nonlinear transformation follows a linear transformation.
So strikingly, our data reveal how flies and vertebrates, separated by hundreds of millions of years of evolution, have converged on a similar computation for generating direction selective signals, a finding that raises additional interesting questions. For example, why do these neurons implement this particular computation instead of some other? And what is its biophysical basis? Hopefully these questions will be the subject of future studies in the field.
Thanks for watching, and we hope you enjoy our paper.